Morning. So for today class, okay, we will learn about protein synthesis, okay, which is also means the protein making process. Okay, see the next slide. Okay, proteins. Okay, proteins make up all living things. Okay, all living materials. Okay, for example, like hair and your nails. Okay, a protein called alpha keratin. Okay, forms your hair and fingernails, and also is the major component care of the feathers, care wool, clothes, scales, care horns, and hoofs, care for the animals. Okay, so for muscles, care muscle protein called actin and myosin can enable all the muscular movement. For example, like from blinking, okay, blinking your eye, okay, to breathing to roller blading. Yeah, cellular messengers, receptor proteins start outside of the cells, okay, and transmit signals to partner proteins on the inside of the cells. Okay, antibodies. Okay, antibodies are proteins that help defend your body against foreign invaders, okay, such as bacteria and virus. Okay, for blood, okay, the hemoglobin, okay, hemoglobin protein, okay, carries oxygen, okay, in your blood to every part of your body. Okay, brain and nerves. So ion channel proteins control brain signaling, okay, by allowing the small molecules into and out of the cells. Okay, for enzymes, your enzymes in your saliva, stomach, and your small intestine are proteins that help you get to digest the food. Okay, the last one will be the cellular construction worker. Okay, so huge cluster of proteins form molecular machines that do your cells heavy work, such as copying genes during cell division and making new proteins. Okay, so you can see that all of these are related to proteins. So which basically means that protein is one of the major nutrients in our body. Okay, so we need protein to survive. Okay, the next slide here. Okay, proteins are composed of amino acids. There are 20 different amino acids. Okay, so different proteins are made by combining these 20 amino acids in different combinations. So this is the chain of amino acids. So at the end here, it become bigger and bigger. So it becomes protein molecule. Okay. Next slide. So proteins are manufactured, okay, which is made by ribosome. The ribosome you can find in the animal cell, okay, normally animal cell and also plant cell. Okay, this ribosome is normally attached to the rough endoplasmic reticulum. Okay, and then next slide here. Okay, so the main function. Okay, the main function of proteins. Okay, the first one is to help fight disease. Okay, different. Okay, any kind of disease. Okay, then to build new body tissue. Okay, when you injured. Okay, so the new cell, okay, will be produced. Okay, so enzymes used for digestion and other chemical reactions are proteins. Okay, what are enzymes? Enzymes speed up the rate of reaction. Okay, it's a substance that speed up the rate of reaction. Okay, and the last one, the main function of proteins, okay, is, okay, proteins are a component of all the cell membrane. Okay, so nucleic acid. So there are two types of nucleic acid in the nucleus of cell. The first one here is DNA, deoxyribonucleic acid. Okay, DNA is like a master program, okay, which control everything in the organism, including production of proteins. Okay, and then you have RNA. RNA stands for ribonucleic acid. So basically, it helps DNA to do their job. Okay, so there are three types of RNA. Okay, the first one will be transfer RNA, okay, tRNA, messenger RNA, mRNA, ribosomal RNA, so rRNA. Okay, so this one is the example. Okay, this one is a messenger RNA, ribosomal RNA, transfer RNA. Okay, the next slide here is talking about the messenger RNA, okay, or mRNA. Okay, so remember that in RNA there is no thymine. Okay, thymine, if you still remember the nitrogen containing bases. Okay, so in RNA there is no thymine, so adenine match up with another base called uracil. Okay, U. Okay, remember that. Okay, please remember that. Okay, so messenger RNA, okay, the function is to copy, okay, the coded message in DNA for specific proteins. Can carries the message, okay, from nucleus to ribosome, okay, where the protein synthesis or production of proteins take place. Okay, function of mRNA. So what is the coded message? So it's called it is called a codon and is made of three nitrogen bases. So each amino acid has a specific codon. Okay, for example, like AAU, okay, is for one type of amino acid. Okay, CCG, one type of acid. Okay, amino acid. Okay, SG is one type of another amino acid. Okay, so that's why here it say each amino acid has a specific codon. Okay, the next slide here. Okay, now we go to the making proteins. Okay, step one, there will be transcription. Okay, remember that. Okay, always start with transcription. So making a protein. So the first step is to copying a genetic information from DNA to RNA called transcription. Okay, and then why? Okay, DNA is a genetic code for the protein that needs to be made, but proteins are made by ribosome. Okay, ribosomes are outside the nucleus. Okay, in the cytoplasm. Normally after transcription, okay, so after the which means after the mRNA is already produced, okay, so this mRNA will go outside. Okay, we go outside the nucleus. Okay, okay, here it says DNA is too large to leave the nucleus. Okay, because we know DNA is double-stranded. Okay, too large. Okay, but our RNA can leave the nucleus. Okay, because RNA is only single-stranded. Okay. Okay, so part of DNA temporarily unzip. Okay, unzip. This is the DNA. So this is what it means by unzip. Okay, and it is used as a template to assemble complementary nucleotide, okay, into messenger RNA. Okay, so hold on a minute. Okay, so this one. Okay, so this is the template. Okay, so this is the from the DNA and this is the mRNA. Remember, okay, so if this is a template, so when it produces mRNA, so this one will be, okay, it must be complementary. Okay, must be harmonized, okay, harmony. So which means that A will always with U. Okay, why U? Why not T? Because mRNA don't have thymine. Okay, mRNA don't have thymine, so that's why U. Okay, U stands for uracil. A is adenine. So if DNA thymine, so it become A. Okay, so guanine, cytosine, G and C. Okay, mRNA then goes through the pores of the nucleus with DNA code and attached to the ribosome. Okay, so this is the part of the nucleus, so it goes outside. Okay, it goes outside. Okay, and then continue with the process known as translation. Okay, this is the tRNA. Okay, okay, the next slide here. Now we go to translations. Okay, translations. Okay, making proteins. Okay, second step is decoding of mRNA in a protein into a protein is called translation. Okay, tRNA, okay, which is known as transfer RNA, it carries amino acids from cytoplasm to the ribosome. Okay, this is the tRNA. Okay, this is the tRNA, this is the mRNA, and you have the ribosome here. Ribosome. Okay. Okay, these amino acids come from the food we eat. Proteins we eat are broken down into individual amino acids and then simply rearranged into new proteins according to the needs and direction of our DNA. So basically we can get the protein that we eat, okay, the food that we eat, okay, for example like milk, fish, cheese, okay, and meats, okay, other meats, okay, a different type of meats, okay, and so on. Okay, a series of three junction bases in an mRNA molecule codes for a specific amino acid, which is called a codon, or other name is triplet code. Okay, triplet code. So each tRNA, this one, okay, has three nucleotides, is CGG. Okay, CGG. So that are complementary to the codon in mRNA. Okay. Remember, this is a tRNA. This is known as anticodon, and not the codon. Okay, anticodon. So which will be complementary to the codon in mRNA. Okay, later on we will look what it means by this one. Okay, so remember each tRNA will carry a different type of amino acid. Okay, so each tRNA codes for a different amino acid. Okay. Okay, let's see the translation process. Okay, mRNA carrying the DNA's instructions and tRNA carrying the amino acids meet in the ribosome. Okay, so the first one here, so we have the mRNA is transcribed, okay, in the nucleus. After the process, it will goes out, okay, through the hole from the nucleus and enter the cytoplasm and attach to the ribosome. Okay, this is the ribosome. This is the mRNA that produced earlier. This one. Okay, so tRNA, okay, B, second part. Okay, the mRNA then enters the cytoplasm and attached to a ribosome. Translation begins in AUG, this one, AUG. Okay, the start codon. So each transfer RNA, tRNA, okay, has an anticodon whose bases are complementary to a codon on the mRNA strand. Okay, so the ribosome positions the start codon to attract its anticodon, so which is part of the tRNA that binds the methionine. Okay, example here methionine. The ribosome also binds the next codon and its anticodon. Okay, so you have two tRNA here. Okay, so this is the codon 1, 2, 3. Okay, remember that 1, 2, 3 cannot be 2 or 1. Okay, must be triplet. Okay, AUG stand for one amino acid. Okay, this one UUC. Okay, different one. So the T1 tRNA will first bind the first one first. Okay, the first here AUG first, and then the second one will bind to this one UUC. Okay, the which is the tRNA, okay, which I meant, okay, tRNA will bind to the second, this is the second codon here. Okay, remember that each of the tRNA will carry a different type of, okay, amino acid. Okay, this is the amino acid, okay, methionine. Then the second one will be different, phenylalanine. Okay. Okay, the next one here, the amino acids are joined together to make protein. Okay, the polypeptide. Okay, the third part here, which is also known as assembly line. The ribosome joins the two amino acids, here methionine and phenylalanine, and breaks the bond between the methionine and its tRNA. Okay, so the first one will go away, okay, will fly away, okay, from the ribosome, allowing the ribosome to bind another tRNA. So the tRNA will keep coming and coming, okay, as the ribosome keeps moving along, okay. So the ribosome moves along the mRNA, binding new tRNA molecules and amino acid. Okay, you can see this one, the third one here, UUU. Okay, so it's also like a different type of amino acid, lysines. Okay, lysines. Okay, so at the end, completing the polypeptide. Okay, so you can see this the different type of amino acid. Okay, which is already bonded together. They will form into polypeptide. Okay. So the process continues until the ribosome reach one of the three stop codon. Three stop codon, which means UGA. Okay. So the result is a complete polypeptide. Okay. Okay. So this one is showing you the triplet code. Okay, with different uh, triplet code with their own amino acid. Okay. For example, like UU phenylalanine. Okay. This one CUU leucine. Okay. CCU proline, okay, which okay, they have triplet one or with their own triplet code. Okay, we have a different type of amino acid. Okay, again, this is example. Okay, just extra information. Okay, no need to memorize. Okay, remember after the end of this slide, okay, you should know protein synthesis. Okay, start with DNA and then undergo transcription, followed by okay and then transcription, then will produce mRNA, and then mRNA will undergo translation, then protein will be produced. Okay, so this is the last one, control of the genes. So each cell in the body contains a complete set of genes, but many of them are switched off, which is also means not expressed. Okay, so they do not produce the corresponding protein. Okay, what it means by, okay, example like the genes to make insulin are found in both the liver and islets of Langerhans cells, okay, which is in the pancreas. Okay, but liver cells do not produce insulin. So the insulin making genes in the liver cells are not expressed. Okay, but okay, the islets of Langerhans cells, however, can control when to produce insulin. So they can control when to switch on or off the insulin making gene. Okay, so in summary, okay, in short, different cells express different genes. Okay, okay, when things go wrong, okay, which is known as mutations. Okay, this part, okay, let's say we a person with healthy hemoglobin. This part is a person with not healthy hemoglobin. Okay, for the wild type hemoglobin DNA, so it should be CTT. Okay, it should be CTT. Okay, but let's say undergo mutation, with this one at the middle is changed into a CAT. In the DNA, the mutant template strand has an A, where the wild type template has a T. So let's say it undergo transcription, so it will produce mRNA. Okay, complementary. So of course it will become G and C, right? TA and TA. Okay, but let's say mutations, okay, happens here. So this still the same, but the middle one is the one who changed. Okay, now it become U. Okay, the mutant mRNA has a U instead of an A in one codon. Okay, and then at the end, during the protein synthesis, okay, protein synthesis, which is the translation. So the normal hemoglobin, so the mutant sickle cell hemoglobin has a valine instead of a glutamic acid. Okay, so this one is related to this one. So sickle cell anemia is one of the genetic disease. Okay, a person who has a not healthy hemoglobin. Okay, the function of hemoglobin, you know that is to carry oxygen okay, throughout the body. Without healthy hemoglobin, okay, so the body will lack of oxygen and eventually okay, the person will die. Okay, okay, so fill in the blanks. Okay, so during protein synthesis, the one strand of DNA okay, is used as a template for the synthesis of mRNA. So this process is called transcription. Okay, the mRNA move okay, from the nucleus get to the cytoplasm, okay, where it attaches itself to a ribosome for the process of translation. Okay, during this process, the ribosome moves along the mRNA. In this way, amino acids are linked to form a polypeptide. Okay, so I think that's all for today's class. Okay, okay, so that's the answer. Okay, so thank you for today.